I'm David Carlson, a graduate student in the lab of James Evans in biochemistry and molecular biology at the University of California, Davis. In this video, I will show you how to build a simple yet effective cryo-light stage for correlative cryo-light electron microscopy. The cryo stage consists of a transparent viewing screen, a cutout for the objective lenses, a liquid nitrogen dewar, a heat sink for viewing your sample, and a sample transfer mount. The majority of materials used were purchased at a local hardware and grocery store. They consist of a 9-inch pie pan, 9-inch cake pan, 4 pairs of chopsticks, great stuff spray foam, a 4 cm by 8 cm by 1 cm block of aluminum drilled with mounting holes for number 10 bolts, 5 number 10 3/4 inch flathead slotted bolts and nuts, 3 number 4 3/8 inch round slotted bolts and nuts, 15 number 10 washers, 2 transparent plastic clipboards, water, and a cryo-EM grid box holder and handling rod, which can be purchased from Ted Pella. Place the pie dish on the microscope stage under the objective lenses. Place the aluminum block near the edge of the dish and focus on the surface of the aluminum. Check that the objective lenses can freely swing in and out over the rim of the pie dish. If not, reposition the aluminum block and check again. Once the location of the aluminum block is determined, mark the position of the mounting bolts with a marker. Position the cryo grid box next to the aluminum block and with a marker, record the position of the notch and each side of the cryo grid box. With the layout of the bolts recorded